Hi everyone, I'm Kelsey and I'm going to be narrating our project that I did with my partner Sushil about how to predict which plants are going to become invasive in Hawaii. And by the word invasive plants, we're referring to any plant that is native to somewhere else in the world, has been brought to Hawaii, and has now escaped to become a permanent part of the vegetation. However, many people in Hawaii are not aware that they're living in a global invasive plant hotspot. So for instance, if you Google images for Hawaiian plants, you come up with the bird of paradise, which is native to South Africa, and plumeria, which is native to the West Indies. And in fact, the ratio of wild-growing alien plants to native species in Hawaii is one of the highest in the world per land area. Actual Hawaiian plants, like the two you see at the bottom right of the screen, are often really hard to find and usually occur nowhere else on Earth. And part of the reason for this is just because there are tons of invasive plants now in Hawaii. They cover 63% of Hawaii's land area and can sometimes have negative impacts, like affecting soil stability, like you see in this picture in the center, that causes landslide. They can smother and kill other trees. They can promote wildfire, affect water quality, things like that. And because the vast majority of plants are introduced to Hawaii on purpose, experts recognize that they needed some way to predict which ones will invade, because not all plants from outside of Hawaii are going to become invasive. And so about 20 years ago, the Hawaii Pacific Weed Risk Assessment was born. This system tries to assign a high risk or low risk assessment category to each species based on 49 questions about their biology. Answers to these questions help them calculate a score, and if that score is over 6, it's listed as high risk, and if it's lower than 1, then it's listed as low risk. So for instance, the plants that I used as example of invaders are rated as high risk with set scores of 17 and 14, and the other ones that I looked at on the last slide are listed as low risk with scores of -3 and -6. However, there is this gray area of recognized inaccuracy between the scores of 1 and 6, where the weed risk assessment can't assign it a high or low risk status, and it instead assigns a category of evaluate, which means that more data is needed. The plant undergoes a second screening, which was developed to improve their accuracy later to see if it can be assigned to either a high or low risk category. However, more often than not, it just kind of stays in the evaluate category and can't be further assigned a status. Ensuring the accuracy of the weed risk assessment system is important because millions of dollars are being spent annually to help stop plants at the border of Hawaii and also conduct invasive plant eradications within its borders. And also, this system has been adopted in many other countries across the world. So specifically, we'd like to ask, how well does the current weed risk assessment program predict plant invasion, since this has not been assessed since it was implemented? And which questions are the best predictors of invasion? And are the importance of these questions given more weight in the manual scoring system? And number 3, can we apply any machine learning models to increase its accuracy? So we collected data from 2 sources. The Hawaii Pacific Weed Risk Assessment dataset was our primary source of data, which included assessments from 1,978 species, as well as answers to the 49 questions, the scores, the ratings, such as high risk, low risk, etc. We also included a list of plants that have escaped cultivation from the Bishop Museum. This list allowed us to compare the prediction provided by the weed risk assessment and the predictions from our machine learning models to actual documented escapes in Hawaii. Unfortunately, almost all of the data was available in just 2 formats: individual PDF files and Excel files for each species. So our first goal was just to scrape the data out of those files to get a single CSV file. So we used 2 different tools, Tabula to scrape the PDFs and XLRD to scrape the Excel files. The files were in a predefined format with slight inconsistencies, but we managed to scrape most files with the help of Python script and then manually adjusted a few of the inconsistent data. Although we didn't have a lot of lines of data to deal with, it was very messy. So some problems we faced were that some of the columns were misaligned during the data scraping from the PDFs, like I just mentioned, and had to be re-entered and verified by eye. Also, the data that was reported in the original Excel files and PDFs was sort of inconsistent. So if they wanted to denote yes, they might have a capital Y or a lowercase y or a Y space or the full spelling of yes, etc. So every column had to be combed through for unique values. Another large hurdle is that scientists often change the name of a species as they learn more about its evolutionary history. So the species names used by the weed risk assessment and the second data set from the Bishop Museum weren't always the same, even though they were referring to the exact same organism. So we had to use a web online application called the Taxonomic Name Resolution Service to update both data sets to the newest acceptable name so that we can merge the data sets. But even then, there are unresolved names that had to be fixed by hand. And ultimately, since there's not a lot of known about each plant species, all of the weed risk assessments had missing data. So we couldn't just delete lines where there were missing data, and we're still dealing with and exploring different options to deal with that. When at last we were able to explore and analyze our data, we were able to address our first question: how well does the weed risk assessment and its manual scoring system predict plant invasions? And the answer is pretty well. It turns out that it only gets it wrong about 10% of the time. So for instance, it inaccurately labeled 135 out of 1,988 species as low risk that have actually escaped from cultivation, according to the Bishop Museum. Additionally, 68 species fell into that evaluate category, only to later escape cultivation. To answer our second research question, which questions asked by the weed risk assessment are most associated with invasion, we conducted a chi-square independence test. So this test allowed us to see which questions are more frequently observed in conjunction with a plant who has escaped cultivation than we would expect if there was no association. This also allowed us to calculate a p-value, with lower p-values indicating a higher probability of association between the weed risk assessment question and its escape status. Then we can rank the questions according to their p-value to assess their relative importance, and we've taken the log of these values to display differences in p-value among our questions. I'll go over an example of questions that were ranked in the top 5 in just a minute. We also use this chi-square independence test method to look at how the weed risk assessment questions were associated with each other, and the log of the p-values are visualized on a heat map of pairwise comparisons shown here. This is important because we'd like to know whether some biological traits are represented by multiple questions and are being double counted in the manual scoring system or in our predictive models. We found that in general, most questions were relatively independent, although there are a few questions that are correlated or appear to be correlated, but there wasn't an obvious biological reason why they should be correlated. We then just tried to roughly compare the top ranking questions to how they were weighted in the manually scored weed risk assessment. So most of the questions are scored as either 0 or 1 based on whether the assessors answered yes or no, but other questions carried more weight, allowing for a larger range in scores of up to 5 points, which presumably means that the biologists who created the weed risk assessment thought it was a pretty important trait. So not surprisingly, our top ranked question was whether the plant was known to have escaped cultivation elsewhere in the world. It's not about to suddenly behave when it gets to Hawaii, and the biologists seem to know that, allowing for a range of scores from up to 0 to 4 points. Our other top-ranked questions were whether it impacts natural systems, whether it's likely to be dispersed unintentionally by humans, whether it causes economic impacts to agriculture or forestry, and whether it's easily killed by herbicides. The biologists who created the manual scoring system seem to agree that these are important questions. They were allowed a score ranging up to 2 points rather than just 1. Our third goal was to see if we could train models such as random forest, KNN, and naive Bayes to improve the accuracy over the manually scored weed risk assessment. So we performed k-fold cross-validation and visualized the models with several different parameters so we could see how the training versus test accuracy changed for different parameter values. And we used this to pick the best parameters with the highest accuracy. So for example, the accuracy of the tanning and tress scores here are shown on the bottom left for the minimum sample split size and the parameter criterion for the decision tree classifier model that we ran. So this helped us choose the best value for the minimum sample split size and the parameter criterion for our model. So as you can see on the top 2 graphs, the accuracy curve is highest at 75% for the parameter criterion entropy, the top right one, and the parameter minimum sample split size at about 500. So ultimately, are machine learning models able to more accurately predict which plants will escape when compared to the manually scored weed risk assessment? The answer is not yet. The highest accuracy we were able to achieve was 79% prediction accuracy with the naive Bayes model, although we're still playing around with the parameters for that one. And the lowest was KNN at 70%. So the manually scored weed risk assessment is still the best at about 90% accuracy. Although we're still working on tweaking our models, future things that would be interesting to look into include exploring models that would predict our missing values to deal with our missing data problem, investigating whether the variable scores that are assigned to different questions by biologists in the manual scored system are useful when applying distance-based models, and also we can investigate the second screening process a little more to see how useful it is in sorting species out of the evaluate category. And that's it. Mahalo for listening. If you have any questions, we'd be happy to take them.